Hi everyone. So today we'll start the topic of formation of enamel, and it contains two topics. First is the life cycle of ameloblast and the second one is amelogenesis. Okay. So the life cycle of ameloblast, it has 6 stages. What is it? I mean, like it's the stages ameloblast or the inner enamel epithelium goes through to form enamel. So we will see these stages. So the first two stages, they are called the pre-secretory stages. So there is no actual enamel formation in these two stages. Then the second and the third and the fourth stage, the enamel formation, the actual enamel formation takes place in these two stages. In the fifth stage, there is protection of enamel. So the enamel is basically covered by a layer in this stage. And then the desmolytic layer stage, here the that layer we formed in the previous stage, it will break down the connective tissue surrounding the enamel so that tooth eruption can take place. Okay, so the first stage is called the morphogenic stage. The second is the organizing stage. The third is the formative, maturative, protective, and desmolytic stage. Now, I'll just give you a brief overview of what happens in every stage. So, in the first stage, the morphogenic stage, this is the inner enamel epithelium. Okay, so it stays, there's no changes taking place in this stage. This is the whole thing. This end is called the upper side, it is called the proximal end, and the lower side, it is called the distal end. So what will happen that there are centrioles and the Golgi bodies in the proximal end. But in the second stage, what will happen? The inner enamel epithelium will elongate and these cells will migrate to the distal end. Okay. So this is what happens in the organizing stage. In the formative stage, what will happen? That in the organizing stage also some dentine will be laid. So in the formative stage, what will happen? The ameloblast will grow a Toombs process, and this will secrete the enamel matrix. So basically enamel formation will start in the formative stage. Now what will happen in the fourth stage, the maturative stage, there is already enamel matrix laid down, okay, and some form of mineralization is also present by this stage. There is some form of mineralization also taking place in formative stage. So what will happen is the ameloblast will reabsorb the organic matrix. It will reabsorb around 96% of the organic matrix, and it will pump in more ions to mineralize the tissue. Okay, and these are two forms of the ameloblast which exist in the maturative stage. Now, fifth is the protective stage. So there's basically formation of a layer of tissue over the tooth so that no reabsorption takes place. And in the desmolytic stage, as I explained, enzymes are released so that the breakdown of the surrounding tissue can take place. Okay, now going to the first stage, which is the morphogenic stage. What happens is the inner enamel epithelium cells are short and columnar. So you have to study the structure of the cell. It is short and columnar, okay, and they have the Golgi bodies and the centrioles. I'll explain to you what is happening. So this is the cell, right? It is columnar, it is short. We have these Golgi bodies. This is the proximal end and this is the distal end. Okay. So the Golgi bodies, the centrioles, they are all on the proximal end and mitochondria is distributed throughout the cell. We have this big nucleus. It is in the center of the cell and there is this cell-free zone present in the dental papilla. So there's basically no cells in here. And then there is a basement membrane also present. So this is all written in here. Okay, and there's nothing else in this stage. Now moving to the second stage, the organizing stage, what happens is that what the cell we had, right, it will grow in size. So the cell we had was columnar, now it has elongated, okay, it has grown in size, and all the cell organelles which were present on the proximal side, now they have migrated to the distal side. Now the centrioles, the Golgi bodies, they are all on the distal side. The cell-free zone we had, it has disappeared because the cell elongated. The basement membrane is still there, and because the cell elongated, it induced the dental papilla to to cause differentiation of the odontoblasts. So since the odontoblasts are differentiated in this stage, they will lay down dentine, pre-dentine. The first layer of dentine will be laid down. So this thing, so the inner enamel epithelium becomes longer and begins differentiating. Okay, reversal of functional polarity. What is reversal of functional polarity? It is migration of the cell organelles from the proximal side to the distal side takes place. Okay, cell-free zone disappears. So that zone has disappeared due to elongation. Inner enamel epithelium comes in contact with dental papilla and differentiates into odontoblast. So this thing came in contact with the dental papilla, which induced the differentiation of odontoblast. Towards the end of this phase, dentine formation takes place because now the odontoblasts are differentiated, right? So dentine formation will take place. Now the third stage, which is the formative stage. What will happen in this stage? Dentine formation in the previous stage is essential for the complete differentiation of inner enamel epithelium into ameloblast. So you have studied in the previous chapter, right, that dentine induces the inner enamel epithelium to form enamel. Okay, so now in this stage, since dentine was formed in the previous stage, now it induces the inner enamel epithelium to form enamel. Okay, in order to form enamel, this ameloblast or inner enamel epithelium needs to have this Toombs process. So what will happen? Ameloblasts develop blunt processes called Toombs process. Okay, these are responsible for rod and interrod secretion, so they will produce the enamel from this side. Firstly, the entire enamel matrix is laid and then mineralization takes place. So what is matrix? You can imagine matrix to be like if you have a bowl of noodles. So you can imagine the matrix to be the gravy and then the mineralization is the noodles you have in here. Okay. So they're both very essential, but first the matrix will be formed and then the mineralization will take place. Okay. Now only a small amount of mineralization takes place in this stage. So in this stage only 30% of the mineralization will take place. The rest 70% will take place in the next stage, which is the maturative stage. What else happens in this stage is that the basement membrane will degenerate. So there is no basement membrane present. It was present up to the organizing stage, right? Dentine deposition cuts off nutritional supply from dental papilla and reversal of nutritional source takes place. So this is an important concept. What was happening that up till now we had no dentine or enamel, right? So what was happening that the enamel organ was deriving nutrition from the dental papilla. I hope you remember this from DOT. So this was the dental papilla and this is the enamel organ. So the enamel organ was deriving nutrition from the dental papilla. But now since we have enamel and dentine over here, so nutrition can't go across this. Okay, there is no nutritional supply. So what will happen is the enamel organ will collapse. Okay, the stellate reticulum that was present in between, it will collapse so that this can derive nutrition from the capillaries of the dental sac. So if you remember the structure of the enamel organ we had, right? So this was the thing. We had the tooth gum. We had three major parts. First was the dental papilla, then we had the enamel organ, and this area outside this was called the dental sac. Okay, the dental sac. So now, since we can't supply nutrition from the dental papilla, we will derive nutrition from the dental sac. For this, there are new capillaries that are budding over here, and to decrease the distance between the enamel organ and the enamel and dentine, this stellate reticulum in which was inside the enamel organ, it will collapse. So it will basically collapse the entire enamel organ. Okay, so this is called reversal of nutritional source, which will take place in this stage, right? um Now what will happen in this is the fifth stage, the maturative stage, the fourth stage, sorry. And the organic components are reabsorbed and minerals are pumped in. So what was happening is until this stage, we had this tooth. Just a second, we had this tooth. Okay, what was happening is we had formed the entire enamel matrix. So the all the soup, the gravy, it was laid, and some of the noodles, you can say, some of the minerals that were also laid. Right. Now what will happen? We will enlarge these minerals. We'll make them bigger. So what we'll do is we'll add more minerals. Okay, we'll add like we'll thicken these minerals so the entire thing is covered, and we'll also begin reabsorption of this organic matrix because we don't need this much of organic content, right? We only need 4%. Only 4% of enamel is organic. Remember, 96% of the enamel is minerals. So we need 96% of the crystals and only 4% of organic matrix. So 96% of the matrix of the soup that was laid, it will be reabsorbed by the ameloblast. So what is happening in this stage? Uh, the organic components are reabsorbed and the minerals are pumped in. 50% of ameloblast degenerate. So the ameloblast that it will take now a new form. It will have two forms. For the 5 hours, for 5 hours, it will be in this smooth border stage, and for another 5 to 7 hours, it will be in that ruffled border stage. Okay, and what will happen in the smooth border stage? It will reabsorb all the enamel matrix, and in the ruffled border stage, it will still reabsorb the matrix, but it will also pump in more ions to cause this mineralization. Okay, so this process of switching between smooth and ruffled stage, this is called modulation. Okay, so the organic components are reabsorbed, minerals are pumped in, 50% of ameloblast degenerate. So 50% of the ameloblast will degenerate over here. Tombs processes disappear, obviously. Now they have smooth or ruffled borders. The cells are reduced in size but are still tall columnar. So they also there is a reduction of size of the cells. Okay, the basement membrane is resecreted, so there will be a basement membrane over here. The ameloblasts show modulation, which I just explained. They alter between being smooth and ruffled. Many vacuoles containing enamel matrix are seen, which indicates reabsorption of the organic matrix. So you see these dots, right? So the enamel matrix will be reabsorbed into the cell, into the ameloblast, and they will be reabsorbed in the form of these vacuoles. Okay, now the protective stage. So what happens in the protective stage? Up till now the entire enamel has been formed and it has been mineralized. So now the enamel needs to be protected from the surrounding connective tissue, because these cells contain basically cementoblast, right? So there can be either reabsorption of the enamel, it will break down, or it will lay a layer of cementum over it. Right. So ameloblasts reduce in size to become cuboidal and can't be distinguished from other layers. So what will happen first is these are tall columnar cells, okay, ameloblasts. Now they will reduce into cuboidal form. Okay, now they are reduced in cuboidal and they can't be distinguished from any other layer of the enamel organ. Okay, the enamel organ itself forms a layer over the tooth called reduced enamel epithelium. So you see, this is the green layer was present over here. Now this entire the structure collapses to form this layer over the tooth. This is called reduced enamel epithelium. Okay, this prevents the enamel from coming in contact with the surrounding connective tissue so that no reabsorption takes place. If the enamel is not protected, it will either be reabsorbed or there will be cementum deposition over it. Okay, so this entire process of formation of reduced enamel epithelium is the protective stage. Now the desmolytic stage. What happens in the desmolytic stage is the reduced enamel epithelium releases enzymes dissolving the surrounding connective tissue. So this is the whole tooth and I have this membrane over it. Okay, now it has to erupt. So what will the this reduced enamel epithelium do? It will release enzymes which will dissolve the tissue surrounding the tooth. Okay, so that it can be the eruption can take place. Okay, so the the tooth can move upwards. This helps in eruption of the tooth. Premature loss of reduced enamel epithelium may prevent eruption of tooth, because then there will be nothing remaining to secrete the enzyme and cause the eruption. Now coming to the second topic, which is amelogenesis. So until now we saw what happens with the ameloblast, okay, how they function. But now we will see how the actual enamel formation takes place, the organic matrix formation and the mineralization I was talking about. So it is the formation of enamel by ameloblast. There are majorly two processes involved: organic matrix formation and mineralization. Organic matrix formation, it takes these are the two processes. Okay, first is the organic matrix formation. I'll just highlight this just a second. So first is the organic matrix formation and second is the mineralization. So the organic matrix formation will take place in the formative stage, where the mineralization will take place in the formative stage and the maturative stage. Okay, mineralization will take place 30% in the formative stage and 70% in the maturative stage. So what is, I'll explain to you what mineralization and organic matrix formation is. Imagine I'm making this bowl of noodles, okay? right? So first I'll add in some water. Okay, uh, just a second. First I'll add in some water. Right. I added that in. Now I'll add that packet of masala we have. Right. So this will form the enamel matrix. We have this matrix, this gravy. Right. Now I have to add in some noodles, of course. Just a second. So I had this masala and water. Now I have to add in some noodles again. So that noodles you can call is that is the crystals. That is the hydroxyapatite crystals. These are the noodles, right? So I'll add that in. And then now, so this happens in the first stage, the formative stage. Now we will kind of thicken the noodles. If you understand what I mean, in mineralization, what will happen is in the second stage, maturative stage, what will happen is there will be no new crystals added, but these crystals or the noodles for the analogy, they will be thickened kind of, right? So when they will be thickened, they will cause this entire crystallization. They will harden the enamel, right? So this is what happens. This is just an analogy, and we'll study further what actually happens. So first stage is the organic matrix formation in the formative stage. So what is happening in the advanced cell stage after the dentine has been laid? The dentine induces the inner enamel epithelium to lay enamel first in the organic matrix. So we saw how one layer of dentine was formed that induced the inner enamel epithelium to produce enamel, right? So first it will lay down the inorganic matrix. Ameloblasts develop these extensions called the Toombs process. They are separated from the cell proper by distal terminal bars. So I'll explain you the structure of Toombs process. The processes contain typical granules as well as rough endoplasmic reticulum and mitochondria. Okay. The Toombs process contains a secretory and a non-secretory site, and the secretory site produces enamel matrix. So what is happening is, we had this dentine which induced the enamel formation from the inner enamel epithelium. Now this will grow a Toombs process. So let's study the structure of the Toombs process. This is the Toombs process. It has these terminal bars. Okay, these are basically incomplete septa. They just divide the cell, the Toombs process from the ameloblast. Okay, so this is the terminal bars. Then it has this non-secretory side, and then this is the secretory side, right? The secretory side also has two divisions. So first is the proximal part, and then this is the distal part. So the distal part, it will produce the rods, okay, the enamel rods, and the proximal part, it will produce the interrods. Okay, so this is the structure of the Toombs process. And now enamel matrix starts forming in this stage, in in here. Now after the matrix has been formed, so let's study first the structure of the organic matrix. Organic matrix has amelogenesis and non-amelogenesis. Okay, these are the proteins that will be laid. So non-amelogenous, these are the proteins mentioned in the book in the organs. So there's ameloblastin and enamelin, which help in nucleation and growth of crystals. So what is nucleation? Nucleation is establishing a center. Okay, so these crystals are nucleated over here, and then they will maybe grow. So this is growth of the crystals. Okay, so the starting of the center is nucleation. So the ameloblastin and enamelin, these are non-amelogenous, and also tuflin. It is involved in cell signaling. So all that the induction and everything that is going on, tuflin has some role to play in it. Now the amelogenins, they maintain the space between the crystals. So as I was explaining to you in here, right, that there will be rod formation and interrod formation, right? So there will be some amelogenin, the proteins present in between over here. So there will be some amelogenins present over here, and it will maintain distance between the two crystals. Okay, genes coding for the amelogenins are present on both X and Y chromosomes, so it kind of varies between the sexes. The protein sequence, it varies between the sexes because the genes are present on X and Y chromosomes. And now we'll study mineralization, but before we study mineralization, you have to understand this thing that my entire enamel matrix is formed by now, okay? But where will mineralization start from? So firstly, it will start from the incisal area, okay, the incisal or the occlusal area. But over there, it will start from the rod. So this is the entire area, right? But if I zoom into this area and I have this enamel rod, so in the enamel rod, it won't start from the upper side or from the incisal side. Within the enamel rod, it will start from the dentino-enamel junction. So the mineralization will kind of start from here, right? It will first start in the incisal area, but again from the dentino-enamel junction, one rod will calcify from the dentino-enamel junction, okay? And it will come on in the cervical area later on, the mineralization, okay? So I'll just for a clearer view. So I have this tooth. I'll just draw it in black. So I have this tooth. So where will be the first enamel? If this is dentine, where will be the first enamel rod calcified? So it will start calcification from here. Okay, and then there will be one rod. So first all the rods from here will calcify. They will start calcifying from the bottom, from the dentino enamel junction, and then from over here it will also calcify, but later on. First this area will calcify and then this area will calcify, and over here even this portion will calcify first. Okay, now moving on. Mineralization. So mineralization has two phases. The first phase is called the immediate partial mineralization. It takes place during the formative stage. Okay, so I explained that mineralization will take place in two stages. First is the formative and second is the maturative. So 30% of mineralization will be taking place in this stage. So what happens is hydroxyapatite crystals are released from Tombs process. Okay, uh, so what was happening that we have these in mineralization, nothing else is released. It's just hydroxyapatite crystals. Okay, we had two ends to the Tombs process. So this end will release hydroxyapatite crystals parallel to the rod. Okay, and then this end, the proximal end, it will secrete hydroxyapatite crystal around 65 degrees to the axis of the rod. Okay, and this will be called interrod. So the distal end secretes hydroxyapatite crystals parallel to the rod axis. So this is the rod secreted. The proximal end secretes hydroxyapatite crystals at 65 degrees to the axis, which forms interrod. So this will form the interrod. These hydroxyapatite crystals which are secreted at 65 degrees to the rod axis. Okay. Amelogenesis maintain the distance between the crystals, so this distance will be filled in with some amelogenes so that they keep the crystals apart. One enamel rod is formed by the contribution of four ameloblasts, and each ameloblast forms four rods. So what is this concept, right? You see that one enamel rod, to form this, I will need four ameloblasts. But then this ameloblast, it will also contribute in formation of four rods. So it formed this rod, it formed this rod, and it will also form like one here and one here, another four rods. Okay, so just another example of understanding this is, you have two parents, right? So you have father and mother. To form you, the contribution of two individuals are needed, right? But you may also have a sibling. So these two individuals may have also contributed to the formation of two people. So one is you, one is your sibling. So this is similar kind of concept. Instead of two, we have four individuals working over here. Okay. Now, phase two. What happens in the second phase of mineralization? So remember, in the first phase, we just laid down hydroxyapatite crystals. We laid one form of crystals parallel to the rod axis, and we laid another crystals at 65 degrees to the rod axis. Okay, so we laid rod and interrod. Now the main mineralization, the 70% of mineralization will take place during the maturative stage. And what happens in this phase is mineral content of the enamel has to be increased. So we are not pumping in more crystals, we are simply increasing the crystals in size in this stage. No new crystals are deposited. The already present crystals grow in size. Okay. 50% of the ameloblast undergo apoptosis, that is cell death. Others undergo transition stage. So what is transition stage? These changes, right? These changes which will take place in the ameloblast. This is called the transition state. Ameloblasts lose their Toombs process, shorten, and show modulation. So this is the transition state. Ameloblasts will ruffle border, absorb the organic matrix, and also pump out the minerals. So I already explained a number of times that these cells will show two forms. The ameloblast, we had the ameloblast. So firstly, in the first stage, it was short, then it grew in size, then it grew a Toombs process. Now it has lost that Toombs process, and now it alters between these two forms. Okay, one being ruffled and one being smooth bordered. So what is happening is ameloblasts with ruffled border absorb the organic matrix and also pump out minerals, increasing the size of hydroxyapatite crystals and mineral content of the enamel. So we had this matrix laid down, we had these crystals. Okay, now what is this ruffled border cell ameloblast is doing? It is in pumping in more ions to increase the size of the crystals, and it is also absorbing this organic matrix inside there. Okay, ameloblasts with smooth borders are relatively less active and they only absorb the matrix. So they are not pumping in any more ions. They are not doing that. They are just absorbing the organic matrix. So and every 5 to 7 hours they uh, change their state. Okay, this will continue until 96% of the enamel is mineralized. So enamel is 96% mineralized tissue and 4% organic. So until 4% of this, only 4% of this matrix remains, and 96% of the enamel is mineralized, this process will continue. Now, and the last topic is why is amelogenesis unique? Because amelogenesis is not similar to any other tissue we have, any other hard tissue we have. So there are several differences in amelogenesis and other tissue formation like dentinogenesis, and we'll study that. So in amelogenesis, the secretory cells, the inner enamel epithelium or the ameloblast, they are of epithelial origin, whereas for other hard tissues, they are of ectomesenchymal origin. So what this means is that we studied the enamel organ, right? Uh, so the inner enamel epithelium, it was made from this enamel organ, which was epithelial in origin, okay? But then the dental papilla, which had odontoblast, it was ectomesenchyme, ec- ectomesenchymal in origin. So the odontoblasts, for example, are derived from ectomesenchymal origin, whereas inner enamel epithelium, it is the only cell, the forming cell, which is epithelial in origin. Okay. In all other tissue, collagen forms a major part of the matrix, whereas there is no role of collagen in amelogenesis. So we don't see any form of collagen in formation of enamel, whereas in every other tissue, every other hard tissue, collagen has a major form in the matrix. It's like aloo; it's present in every sabzi. Okay, collagen. In enamel formation, even the matrix is partly mineralized, but it is not mineralized in any other hard tissue. So we had some crystals of hydroxyapatite in this stage as well, where the matrix was forming. We already had some mineralization present in that stage, but in any other tissue that won't happen. First, the entire matrix will be formed, and then complete mineralization will take place. The matrix is never partially mineralized in any other hard tissue. Okay, the secreted matrix is not reabsorbed in any other hard tissue, but 90% of the enamel matrix gets reabsorbed. So once the matrix is formed, it will never be reabsorbed in any other tissue. But over here, it is unique that the matrix is first laid down and then it is reabsorbed. Okay. In in other hard tissues, the forming cells, that is the odontoblast, are present throughout the life. But in case of enamel, the ameloblast undergo apoptosis. We saw that 50% of ameloblasts were dying and then they were forming reduced enamel epithelium. So that won't happen with any other like odontoblast and everything. They will stay there throughout. So they will form secondary dentine or tertiary dentine. They will stay with you forever in throughout your life. But the ameloblast, they die. Okay, they don't stay there. This is the only forming cells which die. Okay, so this is it for the topic. Hi everyone. So this is the last video on the topic of enamel, and we'll be talking about the 8 changes of enamel and some clinical consideration that comes along with enamel and the formation of enamel. Okay, so starting with the 8 changes of the enamel, what happens as we age? What changes can we see in the enamel? So the first thing that happens is the wearing off of the occlusal and proximal surfaces due to mastication. What that means is that when the enamel is young, so you can see the smooth and maybe the cusps are pointed, that kind of surface. But as we age, due to mastication, the surface will be worn off. Then the second thing is the disappearance of rods. So what that means is that we have seen that not disappearance of rods but disappearance of rod ends. So we saw in the structures that there were like rods end on the surface of enamel. So due to the wearing off, they will be also removed. Okay, as the enamel surface gets worn off. Flattening of perikymata. So perikymata were these layers. They were the striae of retices on the surface of enamel that were visible. So they will be also worn off. Facial and lingual structures lose their structures more rapidly than the proximal surfaces. So the proximal surfaces are not affected a lot by mastication. So that's why they won't lose the structures a lot. Anterior teeth lose structures faster than the posterior teeth. It's because the anterior teeth come in the mouth first. Okay, so they will start losing the structures first as well. Localized increase of nitrogen and fluorine in the superficial layers. So as we age, the content of nitrogen and fluorine will increase in the superficial layers. Organic portion near the surface becomes darker and its resistance to decay increases. So you may have heard that enamel becomes harder as we age. So it does not become harder, but its resistance to decay increases because every organic portion it becomes kind of more resistant to bacteria and everything surrounding it. Okay, permeability decreases due to increase in crystal size from the ions acquired from oral fluids. So what's happening is we had this hydroxyapatite crystal. Okay, so we have like we had like 2 of these crystals. So there are pores in between these crystals. Okay, so this allows permeability. But as we age, what's happening is these crystals are increasing in size. Okay, they are gaining more inorganic content. It's accumulating over them from the saliva. So as they increase in size, now there is no pore. This pore, I think, disappeared. So this pore has gone away. That was providing the permeability. So this, because of this, the permeability will decrease. Okay, and it thickens due to the ions acquired from the saliva and the oral fluids. Now coming to the clinical considerations. So what do we have to take care of in case of enamel? So during cavity preparation, no unsupported enamel rod should be left. It may break, produce leakage, inducing secondary caries. So what will happen is, so I have this tooth, okay, and I'm forming a cavity. So if this is the direction of the enamel rods in this portion, so if I'm forming a cavity, if I'm making a cavity, I'll have to make sure that I make the cavity along this, okay, like this. If I try to make a cavity like this, what will happen is that these rods, if I made a cavity, just a second, then these rods will be left unsupported. Okay, let's say I made a cavity over here. Now, these rods over here, they are left unsupported and they might fracture or produce leakage. Okay, so we have to take care of the direction of the rods and make a cavity accordingly. This might also induce secondary caries because if this breaks down over here, there will be a gap and there will be passage for entry of bacteria. Okay. Deep enamel fissures and dental lamellae predisposed to caries. So it's a similar kind of concept here, leaving like a gap. There's a huge organic content in here. In dental fissure, there is empty space, and in dental lamellae, there is space filled with organic content, which allows bacteria to enter the tooth, and it causes caries. Dental caries in children can be reduced up to 40% through the application of sodium or stannous fluoride. So we all know that fluoride reduces the caries. So sodium or stannous fluoride, it can reduce caries up to 40%. Okay. Now moving on to clinical consideration in amelogenesis. So what are the defects caused during amelogenesis, and what conditions can arise? So there are 2 processes in amelogenesis. First is the enamel formation, and second is the maturation. That is the mineralization. So if there is a defect in enamel formation, then hypoplasia will occur. And if there is a defect in maturation, then hypocalcification will occur. So first we'll see what is hypocalcification. So as the name suggests, hypocalcification means low mineral content. Okay, it is there is less mineralization. Why does it occur? So it appears. So first we'll see what it looks like. So it appears like a opaque chalky area on the surface. Okay, The example of hypocalcification is mottled enamel. You can see that mottled enamel over here. A deficiency of mineral content in the enamel is seen. So there is low enamel content. The enamel is soft and acid soluble. The surface appears dull and opaque. It gets discolored and starts getting peeled off in there. So the enamel is weak. It is soft because there's low calcium content. It appears dull and opaque, and there's discoloration. And when you masticate, it will get peeled off. Mottled enamel is an example because due to high fluoride content, the high fluoride content it prevents calcification of the enamel. Okay. So this was during mineralization, and what will happen when the amelogenesis is affected during matrix formation? That is hypoplasia. So what happens in hypoplasia? Pitting, furrowing, or a complete absence of enamel. So all these three structures, all these three conditions are example of hypoplasia. Okay, it can occur during three conditions. It can be systemic, it can be local, or it can be genetic. So what is systemic? It is due to nutritional deficiency. When the enamel in your water, when the fluorine in your water is less than 1 parts per million, it can occur. Okay, so it is due to environmental factor, systemic one. It is called chronological hypoplasia in case of systemic. It appears as a narrow band of defective enamel because what's happening is your enamel is forming, right? So maybe for this period of 1 year, you were having water that had low fluoride content. So this portion of the enamel, it was hypoplasic. Okay, so it appears as a narrow band of defective enamel due to metabolic disturbances because they generally persist for a short duration and the disturbance will go away. Okay, so you can see this band kind of. So maybe the person had initially they had low fluoride content, so it's a mild condition. Local. So what in local hypoplasia can occur due to infection to the pulp. So if this is the pulp and this is the tooth, so maybe there was an infection over here. So this area will be hypoplasic. Okay. Now genetic. So in genetic condition, it is disturbance of the so the entire ameloblast, okay, that is affected. So all the teeth and the entire enamel is affected. Teeth appear yellow, brown, glossy, and hard. Okay. So they will be yellow, and even in this they have bands, but the entire teeth will be like yellow and hard. The anomaly is transmitted as in dominant character. So remember that it is a Mendelian dominant character, which is quite interesting to read in the book. So there are not many questions asked on these topics, at least on the clinical considerations, but you can always add that on for the extra marks in the university exams. And so we have completed the second chapter. If you like videos, please like and subscribe.